This is Selma Schimmel, and you are looking live at the great city of Chicago, which is once again playing host to the American Society of Clinical Oncology, ASCO. This is ASCO's 49th annual meeting, and this year's theme could not be more appropriate: Building Bridges to Conquer Cancer. More than 30,000 of the world's foremost cancer specialists are here, and so is the Group Room, making our 15th appearance at ASCO and one of our very best. Joining me now is Dr. Stefan Gluck, professor in the Department of Medicine, Division of Hematology Oncology at the Sylvester Comprehensive Cancer Center, University of Miami, Leonard M. Miller School of Medicine. Hello, Dr. Gluck.
Hi, thank you very much, Selma, for having me again.
You are one of the leading forces in the area of breast cancer and breast cancer research, and specifically today, we're going to talk about some of the novel drugs that are available for patients with metastatic breast cancer that express HER2.
Yes, it's a very peculiar subtype of breast cancer, and it's one of the things that we learned over the last couple of decades. Breast cancer is not breast cancer, is not breast cancer. It's not only stage different and different in size and in features that the pathologist can discover, but it has actually a very distinct, different molecular structure. And initially we understood for many years, actually for over a century, that estrogen plays a role. Later we also learned the estrogen partner, progesterone receptor, and more recently, about 20 years ago, through the UCLA group, HER2. HER2 is abbreviation for Human Epidermal Growth Factor Receptor number 2. So if there's 2, there must be others, and there are indeed 4 such receptors. This particular marker, molecular marker, has one feature, and the feature is if activated, if expressed, if amplified, so if present in an active form in cancer cells, it makes this type of cancer extremely aggressive, fast-growing, liking to metastasize, causing trouble, causing angiogenesis, and be unresponsive to estrogenic pathways. So our tamoxifen and similar drugs don't work in these cancers so well. With monoclonal antibodies, such as trastuzumab, better known as Herceptin, we can target these cells. And combining with other agents, such as estrogenic agents, we are making major progress. So you asked about the progress in this subtype of disease. As we learned, Herceptin works well, but not alone. Standalone Herceptin is a good drug, but in combination with other drugs, it actually is much better and shines. We have efficacy up to 70–80%, which is in breast cancer unheard of if we combine Herceptin with other compounds. Now, ultimately, unfortunately, most, not all, most of women who have HER2-positive breast cancer and are treated with trastuzumab, Herceptin, actually will have progression. So the cancer becomes resistant, and our treatment failed for her. What it means that we need to have new compounds, new approaches, and maybe 6 years ago, another drug, lapatinib, was tested and approved, which is a second drug targeting HER2. It also targets the first of the 4 components. And then we have many, many more, and 2 of those were actually approved during the last 12 months in the United States and many parts of the world. One is called T-DM1, and the other one is called pertuzumab. Okay, so T-DM1, just to explain to our viewers and people who are interested, is actually a very novel approach how we construct drugs or compounds or treatment entities, I must say. T-DM1 is the abbreviation for 2 things. T is trastuzumab, Herceptin. So it actually has the body of Herceptin. It's a monoclonal antibody, so it's like a Y form, which is attaching to the HER2 molecule. But the T-DM1 is also DM1, which is a maytansine, which is a chemotherapy compound discovered in the 60s, believe it or not. And it was tested in MD Anderson phase 1 studies. It was so toxic and so effective, but the toxicity actually led to death of some patients. So the drug was shelved. But it was so effective that some smart scientist has said, what if we can get this very toxic drug, toxic to the cancer cell, only into the cancer cell? So the idea was to combine these 2 things. But combination of drug and antibody is not easy, so they had to construct something what now is called linker. It links the 2 drugs together and to such a way that the drugs are completely attached to each other and do not do any harm or almost no harm in the circulation when infused.
I have just a curious question. So a drug is developed very toxic. It gets shelved, not canceled. Has it been pulled off the shelf until now?
Yes, it has been sitting on a shelf for 35 or 38 years.
And it's the first application it's been pulled. How did someone come to think to pull it off the shelf for this?
Yeah, I think in hindsight it's easy to understand and logical. What the scientist says, if we had a very effective drug, doesn't matter how toxic it is, and we attach it to something which targets only the cancer cell, HER2 positive cancer cell, hence not targeting anything else in the body, we will have this compound attached to it, this toxic compound, maytansine, and it will target only the cancer cell. Now there's one thing that I didn't tell you yet is when trastuzumab, Herceptin, attaches to the cancer cell, it is being drawn inside the cell. It's called internalized. So it comes into the cell and does it work. Now, and here comes the trick. Within the cell, the linker is degraded and a toxic chemotherapy drug, maytansine, is released within the cancer cell and kills the cancer cell.
Does that mean it does not go systemically?
That is absolutely correct. So although it's circulating in the blood, it attaches to the HER2 cell and goes inside.
So that's really brilliant.
That's brilliant. And you know, just to say some side effects. Side effect of chemotherapy we all know is hair loss. People who have TDM1 infusion do not get hair loss. Chemotherapy induces nausea. They are not sick. They do not vomit. Chemotherapy drug reduces the white cells. The white cells are not being touched. This chemotherapy drug, which is highly effective and extremely toxic, loses its toxicity outside the cancer cell and keeps the efficacy inside the cancer cell.
Do you see that this is a possibility that this will have efficacy for other cancer types?
Yes. So the name Kadcyla comes from the ADC core. The ADC is antibody drug conjugate, and that's the idea. So any antibody targeting something with any drug, cytotoxic, chemotherapy, combined together is the principle. So you tell me you have a target for lymphoma, CD20. Rituximab is such a monoclonal antibody. Why don't we put rituximab with some of these compounds with this linker and create a new ADC, antibody drug conjugate? So Kadcyla is the ADC inside with a K in the beginning and YLA at the end. That's the name.
I'm wondering, while it's now being applied to metastatic cancer, do you think at some point it will be a possibility for earlier stage breast cancer?
Absolutely superb idea indeed. First of all, the drug was approved based upon a study which was in so-called second-line metastatic. So patients who had metastatic breast cancer, HER2 positive, had in the past some chemotherapy and trastuzumab, remember I told you before, and progressed on trastuzumab. So trastuzumab lost its efficacy. Were randomized to a combination of two drugs that were approved 6 years ago, lapatinib and capecitabine, or the other half of the patients were randomized to receive the new drug, TDM1. And there was a significant and clinically meaningful difference in the efficacy. Several months longer progression-free survival, many percentage more response rates, but more importantly, almost no side effects. Exactly what I said before. There's a little bit of hepatotoxicity, which means the liver enzyme can be a little bit higher, it's transient and goes back to normal, and sometimes the platelets can go a little bit low. But that's about it on side effects. And this was enough data with a second analysis that overall survival was also prolonged with the TDM1 that the FDA said, let's approve it.
And no cardiovascular issues?
No more than trastuzumab alone, less than 1%.
Thank you, Dr. Stefan Gluck, Sylvester, Distinguished Professor, Department of Medicine, the Division of Hematology Oncology, the Sylvester Comprehensive Cancer Center, University of Miami, Leonard M. Miller School of Medicine.
Thank you.